In this video, we will understand about how KRAS function. So far, we have understood, we have gone through many papers about the cancers, and one of the common mutations in most of the cancers is this KRAS mutations. So we will understand what is the regular function of this KRAS, it is wild type KRAS, and how it is affected when there is mutation in the KRAS. This KRAS comes from this family of RAS, and RAS has three isoforms, that is HRAS, KRAS, and NRAS. So out of this, in cancer, people have found out the mutations in the KRAS is more common. For example, in the pancreatic cancer patients, around 80% of the pancreatic cancer cases has these KRAS mutations, and these KRAS mutations are usually found in three different positions, that is G12, G30, and Q61. So before going into the mutations, let us understand how this wild type KRAS functions. So in the inactive form of this wild type KRAS is when it is bound to GTP, and once this receptor tyrosine kinase gets activated, then it will cause this GEF to replace this GTP with GTP, and this GTP bound KRAS is the active form of KRAS. It will activate the downstream signaling pathways, which are mainly related to proliferations, differentiation, and survival. However, there is a GAP that is present within the cells, and its function is to hydrolyze this GTP to GDP. So when this GTP is converted into GTP, then the cells will again become inactive, and it is not necessary that GAP is required to convert this KRAS GTP bound state to KRAS GTP bound state. This KRAS also has a self-inactivating ability by cleaving of the terminal phosphate to convert this GTP to GDP. However, that process is slow. So what happens when there is a KRAS mutations? When there is mutations, it usually weakens the ability of this GAP to hydrolyze this GTP to GDP, and because of that, this KRAS will remain in the active form for extended duration of time compared to the wild type KRAS, and this extended duration of times will affect in terms of this proliferations, differentiations, and survival pathways. And since we know the cancers has a property on control growth rate, so this state will support the cancer cells accelerated growth rate, and that will increase the chances of further mutations. So this is how the KRAS functions, and these KRAS mutations are not only found in this pancreatic cancers, but it is found in the other cancers also, and we have seen the results that we have discussed in the previous paper, where they have talked about this non-small cell lung cancers also has the KRAS mutations. So the drugs that is generally that is made in the that we have discussed in the previous video about this RMC 6236 is targeting the multiple mutation of KRAS, and before that what was happening, the drugs were targeting specific mutations of the KRAS to a different amino acid. However, this RMC 6236 unique point is that it can target multiple KRAS mutations, and thereby efficacies of that KRAS mutations is efficacies and also the applicability of those drugs that targets the different mutations of KRAS is increased significantly compared to the previous drug. And in this case, although they are representing the three main signaling pathways, out of which this P13K belongs to the survival pathway, and this NF-kappa beta is also belongs to the survival pathways, whereas this MAPK pathway has a role towards this proliferations or differentiations. So this is what happened when these KRAS mutations are present within the cells. It will cause the cells to accelerate in terms of the growth rate, and that will enhance the chances of further mutations. I hope you understood how this KRAS actually functions and which functions get affected when there is a mutation in the KRAS in most of the cases. If you need to understand more about this concept in detail, you can go to the reference that has been provided in the description. So that's all for this video. Thank you for your time.